Question number 1. A desirable change in genotype of an organism is obtained by A. DNA replication, B. Protein synthesis, C. rDNA technology, D. mRNA formation. First of all, Francis Crick proposed central dogma which states that DNA undergoes replication, which undergoes transcription to form mRNA, which undergoes translation to form protein. This all process of DNA replication, protein synthesis which is translation, mRNA formation which is transcription occurs in vivo. This is the only technology which occurs in vitro. We can carry out the experiment outside the body to obtain a desirable change in the genotype, in the genetic makeup of the organism. So option C is the correct answer. Question number 2. The first human hormone produced by recombinant rDNA technology is option A thyroxine, option B progesterone, option C insulin, option D heat resistant gene. So the first rDNA technology was used in 1978 to produce human insulin with the help of Escherichia coli, a bacteria which is a normal habitant of gastrointestinal tract of humans. So with respect to rDNA technology and Escherichia coli, human insulin was synthesized first in 1978 in United States of America, which was sold under the brand name of Humulin. So option C is the correct answer. Question number 3. Which vector can clone only a small fragment of DNA? Option A plasmid, B cosmid, C bacterial artificial chromosome, D yeast artificial. There is in prokaryotes extra chromosomal DNA is in the form of circular DNA, which we call as plasmids, which codes for specific characters, of bacteria. So this small part or extra chromosomal DNA can be used as a medium to transfer genes and for cloning. So option A is the correct answer. Question number 4. A shape of bacteriophage is. First of all, bacteriophage is a virus infecting bacteria. It is a virus which infects bacteria. So it is of shape of lambda shaped, or when you see it, it is a tadpole shape, which is the shape of larvae of frog. So option D, which states that tadpole like, is the correct answer. Question number 5. The life cycle of lambda-phage is. Lambda-phage is a virus which infects bacteria. So while infecting bacteria, it incorporates its genetic material into the bacteria, which then replicates into the bacteria to form small viroids, which then escape from the bacteria by lysis, So there is this process is lysis. That is why we call this life cycle of lambda-phage as lysogenic cycle. So option C, which states that lysogenic cycle, is the correct answer. Question number 6. Transposons are sequence of DNA, mRNA, rRNA and tRNA. Transposons are sequences of DNA, straight away information. They are also known as jumping genes, and they cannot replicate on their own. So option A is the correct answer, which states that transposons are sequence of DNA, also known as jumping genes, and they cannot replicate. Question number 7. The cloning vector M13 has genetic material. What? M13 is a type of phage but different from lambda phage. It has 72 nucleotides and is DNA molecule is smaller than lambda phage. Is smaller than lambda phage. So this M13 phage has a circular and single-stranded DNA as its genetic material. So option C, which states that single-stranded DNA, is the correct answer, that the cloning vector M13 has genetic material as single-stranded DNA which is circular. Question number 8. In the nomenclature of enzymes of restriction endonucleases, the Roman numeral indicates. So there are three letters, first, second and third. The first letter comes from the genus, the second letter comes from the species of first two letter, from the first two letters of species the second letter comes from, and the third, which is a Roman numeral, indicates the order of discovery from the source, order of discovery from source. So option B, which states that the order of discovery from the source, is the correct answer. Number of times it is used, no. Number of cuts, no. So option B is the correct answer. Question number 9. Gel electrophoresis is used for. Option A, isolation of DNA, B, cutting of DNA, C, separation of DNA fragments according to their sizes or length, and D, constriction of DNA. So gel electrophoresis is a technique. Gel electrophoresis is a technique which is used in rDNA technology when we use recombinant DNA, which we fragment. The fragments are being created with the help of restriction endonuclease enzyme, and these fragments which are being created, which are of various sizes, and these various sized fragments are then separated by this technique of gel electrophoresis. So option C is the correct answer. Question number 10. A gel is used in electrophoresis is. Gel electrophoresis, again, it is a technique which is used for separation of DNA fragments which are necessary for rDNA technology. So the gel which has been used in electrophoresis technique for separation of the DNA fragments is agarose gel, which has been derived from seaweeds. So option A is the correct answer. Question number 11, which states that RNA interference involves. It is option A, interference of RNA is synthesis of DNA, synthesis of mRNA from DNA, synthesis of cDNA from RNA using reverse transcriptase, and desilencing of specific mRNA due to complementary RNA. First of all, this RNA interference is being used in reverse transcription first, but this RNA interference is not for synthesis of DNA, of cDNA, but the RNA which we are using needs to be silenced, or silencing is done of this mRNA by using a complementary RNA, so that it does not get transcript into its specific, or it does not get translate into its specific protein. That is why we need to silence or stop the translation process of mRNA by using a complementary RNA. So option D, which states that silencing of specific mRNA due to the complementary RNA, is the correct answer for RNA interference, which has been used in reverse transcriptase process, and it is required for stopping the translation of mRNA into its corresponding protein which it codes for. So option D is the correct answer. Question number 12. Genes of interest can be selected from a genomic library by using what? First of all, you should understand this, that genomic library is being used for obtaining the entire genome of an organism or total DNA of an organism. So insertion, so what we require from this total DNA can be obtained by treating it with restriction endonucleases. So we get fragments of DNA which codes for the particular proteins, which will code for particular protein. So this DNA, these fragments are nothing but we call them as probes. So these probes are been obtained from the genomic library. So option D, which is the DNA probes, is the correct answer. Question number 13. During the process of isolation of DNA, chilled ethanol is added to first facilitate action of restriction enzymes, no. Second is to for proteins such as histones, no. To remove proteins such as histones, no. Precipitate DNA, C, and D break open the cell to release DNA. So first, when we treat ethanol with DNA being acidic in nature, since when it is acidic in nature, when you treat it with an alcohol, it will precipitate, it will form precipitation will occur of the DNA fragments. So that is why we used chilled ethanol with so as to get the DNA in the precipitate form. So option C is the correct answer. Question number 14, which states the first step in PCR technique is. PCR is polymerase chain reaction. So polymerase chain reaction, there are three steps. First, heat denaturation. Second, annealing. And third, polymerization. So these are the three steps which you can remember as HAP. So the first step is heat denaturation. So option B is the correct answer. Question number 15. DNA fragment generated by restriction endonucleases is a chemical reaction which can be separated by. So gel electrophoresis we use to obtain DNA fragments by separation. So gel electrophoresis has been used. So option B, which states that electrophoresis is the correct answer for question number 15. Question number 16. Which of the following BT crops is being grown in India by the farmers? Bacillus thuringiensis is the full form of BT. So Bacillus thuringiensis is the full form, which is nothing but incorporation of BT toxin gene into the crop. So it releases exo as well as endotoxins. So it prevents from parasites or any insects. So it prevents or protects the crop. So the first crop which was being used with this BT toxin gene was cotton. So option A is the correct answer. Question number 17. The BT toxin, which is Bacillus thuringiensis toxin, BT toxin is Bacillus thuringiensis toxin, is been coded by, it has been coded by a gene which is a toxin which can be exo as well as endo, which has been coded by a gene known as cry gene. And this cry gene is of 4 types, that is 1, 2A, 2B, 3 and 4. So these 2 we collectively term them as 2, which is further divided into A and B, and 3 and 4, which codes for this toxin gene. So option B, which states that cry gene, is the correct answer for question number 17. Question number 18. Cry gene is obtained from. So cry gene which codes for Bacillus thuringiensis toxin. So BT is nothing but Bacillus thuringiensis. So option B, Bacillus thuringiensis, is the correct answer for cry gene. It has been incorporated into crops so as to protect them from insects. So option B is the correct answer for question number 18. Question number 19. USA had taken patent of haldi powder as a wound healing agent in. So USA in University of Mississippi Medical Center of USA had taken the patent on turmeric powder. So had taken the patent on turmeric powder and it was in the year 1995. So option C is the correct answer. Question number 20. In India, research in genetic modification of organism safety issues are controlled by. For turmeric patent against USA, that Mississippi Medical Center, Indian CSIR Research Center had filed case for biopiracy. So this for to obtain the turmeric patent. So it was in India, research in genetic modification organism safety issues has been controlled by CSIR. So option C is the correct answer for this protection of for biosafety issues in India. Is controlled by CSIR. Question number 21. Cultivation of BT cotton has been much in the news. So the prefix BT means Bacillus thuringiensis. So it is from the Bacillus thuringiensis this BT, the scry gene is obtained which codes for the BT toxin. So option B is the correct answer, carrying an endotoxin gene from the Bacillus thuringiensis. So option B is the correct answer for 21. Question number 22. Jumping genes in maize were discovered by. Jumping genes are also known as transposons. So the transposons are nothing but jumping genes which were discovered by the scientist Barbara McClintock. So option C is the correct answer for question number 22, which states that jumping genes in maize were discovered by scientist Barbara McClintock. The first transposons which were discovered by this Barbara McClintock. Question number 23, which states that which one of the following commonly used in transfer of foreign DNA into the crop plants? Trichoderma Hertzium, Myliodogma incognita, Agrobacterium tumefaciens and Penicillium notatum. For it is a pure information that Agrobacterium tumefaciens is a gram-negative bacteria which is rod-shaped. It is a rod-shaped gram-negative bacteria which causes crown gall disease in crop plants, but it has a tumor-inducing plasmid. Because it has a tumor-inducing plasmid, a portion of tumor-inducing plasmid is been incorporated into the host cell, so that tDNA. So this tDNA carries the information for the tumor-inducing plasmid. So to prevent crops from this crown gall disease, it is a commonly used medium from Agrobacterium tumefaciens. So option C is the correct answer. Question number 24. Which one of the following is used as vector for cloning genes into higher organisms? Salmonella typhimurium, Rhizopus nigricans, retrovirus and the babelovirus. Option C is the correct answer because it's a pure information that retrovirus have enzyme known as reverse transcriptase which can convert RNA into cDNA. It can convert this. So the RNA coding a particular protein can be transformed into a DNA sequence into the host. So it is the most commonly used, or as it is used as a vector for cloning genes into higher organisms with more complexity. So there is complexity. So this to tackle the complexity we use retrovirus, which has a reverse transcriptase enzyme which can convert RNA into DNA sequence so that the cloning genes can be easily transferred. Question number 25. Polymerase chain reaction technology is used for A. DNA identification, B. DNA amplification, C. DNA repair, and D. Cleave DNA. So correct answer is PCR, polymerase chain reaction, is been used to amplify the required trait. The required trait is for amplification of the required trait, which has been obtained from DNA amplification, which can be obtained by DNA amplification. So correct option is option B, which states that DNA amplification is the correct answer for the question number 25. Question number 26. Which one of the following palindromic sequence in DNA can be obtained or modified cut at about the middle by some particular restriction enzyme? So palindromic sequence is nothing but which reads same from left to right and from right to left, from on one strand and the other strand. So which can be read by the restriction endonuclease enzyme same whether read from right to left or left to right on one strand and the other. So here in the given options you can see that option B, which when we cut it in the middle, it reads the same from right to left and from left to right if read by a restriction endonuclease enzyme. So option B is the correct answer. Question number 27. An improved variety of transgenic basmati rice is what? First of all, transgenic basmati rice is also known as texmati. So it is incorporated gene. This incorporated gene, it protects the crop. It protects the crop from all type of insects, pests and diseases. So this improved variety, which is transgenic, is resistant to all of these. So option B, which states that is completely resistant to all insects and pests and diseases of paddy, is the correct answer. Whereas rest, which gives high yield and does not require chemical fertilizers, are wrong. So option B, it states that completely resistant to all insect, pests and diseases of paddy, is correct, and texmati was obtained from this transgenic conversion. So option B is the correct answer. Question number 28, which states that genetic engineering has been successfully used for producing what? Option A, transgenic models for studying near treatments for certain diseases, for cow or roses which produce high yield for milk for making ghee, animals like bulls for farm work as they have high powers, or transgenic mice for treating safety of the polio vaccine before use in the humans. So question number 28, genetic engineering is incorporation of desirable traits. So we use genetic material like genes to bring out certain traits. So there is conversion. So this conversion is desirable to bring out the certain desirable change. So conversion, desirable change. So it has been used for the human welfare. So option D, which states that transgenic mice. So we have polio vaccine, which is a live vaccine. So it is live attenuated vaccine. Live vaccine means live viruses are there which are been killed, heat killed. So they are been heat killed. So this vaccine, so if we directly use in humans, there can be certain adverse effects. So first we treat them with transgenic mice. Transgenic mice are the mice which has certain characters of humans as well. So before directly treating into the population, we can treat it with transgenic mice to test the safety of this vaccine. So option D is the correct answer, that transgenic mice for testing safety of the polio vaccine before use in humans. Before using them in humans, this can be used. Question number 29, which of the following are used in gene cloning? Gene cloning is gene replication. So for that we use vectors which can replicate. Vectors are segments of DNA. And in case of gene cloning, we use them as extra chromosomal DNA of prokaryotes, that is in the form of plasmids. So option C, which states that plasmids are used for gene cloning. For gene cloning purpose we use plasmids. So option C is the correct answer. Question number 30. Insect tolerant genes from Bacillus thuringiensis, that is Bt genes, that is cry genes, are being introduced using which vector? So it is a vector which is Ti. So this Ti plasmid is nothing but tumor inducing plasmid, which has been obtained from a bacteria which is gram negative and rod shaped, which we call it as Agrobacterium tumefaciens. So this Agrobacterium tumefaciens Ti plasmid is obtained from this gram negative bacteria, so which helps in incorporation of the desirable gene, so acts as a vector. So this cry genes are incorporated Ti plasmid of gram negative rod shaped bacteria, that is Agrobacterium tumefaciens, to obtain the insect tolerance in the crop. So the correct option or the answer is option B, which states that Agrobacterium tumefaciens is the correct answer for the question, which states that insect tolerant genes from Bacillus thuringiensis, which is Bt, which are cry genes and which are incorporated Ti plasmids of Agrobacterium tumefaciens, and which when incorporated into the host they give us the insect tolerance, also again tolerance against pests. So option B, which states Agrobacterium tumefaciens, is the correct answer.